the Human Genome Project. The human genome has been the focus of biological research for the last 2 decades. Will continue to be the center of attention for many years to come. It is a worldwide effort to identify all human genes of each chromosome along with several other goals. The Human Genome Project is an enormous understanding in genomics that is providing scientists with exciting insight into human genes, their locations and functions. It was originally a 13-year project, 1990 to 2003, coordinated by the US Department of Energy and National Institutes of Health. Additional contributions came mainly from UK, Japan, France, Germany, Australia, China. The aims of the project were to identify all genes, approximately 20,000 protein coding genes in human genome, determine the sequence of the 3 billion chemical base pairs that make up human DNA, store information in databases, improve tools for data analysis, transfer related technologies to private sector, address ethical, legal, social issues that may arise from the project. A major quality assessment of the human genome sequences was published in 2004, indicating over 92% of samples exceeded 99.99% accuracy, which was intended goal. The Genome Reference Consortium (GRC), which is an international collective of academic and research institutes with expertise in genome mapping, is still working on the analysis of the data obtained from the Human Genome Project. The sequencing of the human genome holds benefits for many fields, from molecular medicine to human evolution. Benefits of sequencing of the human genome: improved diagnosis of various diseases, identification of mutations linked to different forms of cancer, the design of medication and more accurate prediction of the effects, gene therapy and control systems for drugs, study of human evolution and anthropology. Another proposed benefit is the commercial development of genomics. Research related to DNA-based products, a multi-billion-dollar industry. The Human Genome Project and the other genome projects such as Escherichia coli, yeast, mouse, Arabidopsis, rice have already been completed. This is therefore opened the way of comprehensive description of molecular activities of human cells. The ways in which these activities are controlled. This is central to the continued development not only of molecular biology and genetics, but also those areas of biochemistry, cell biology, and physiology, now described as the molecular life science. The genome projects will have additional benefits that at present can only be guessed at. It is evident that the human genome, in common with the genomes of many other organisms, contain extensive amounts of intergenic DNA. It was thought that most of the intergenic DNA has no function except occasionally. It is known that some intergenic DNA act as control genes nearby. Could the intergenic DNA have a role, but one that present is too substantial to grasp? Though little is known about them, they are both to have regulatory functions. The first step in addressing this possibility is to obtain a complete description of the organization of the intergenic DNA in different genomes so that the common features which might indicate a role for some or all of these sequences can be identified.